Hello. Welcome to the Deaf Hub Exploring Perspectives. My name is Jenna. I will be the host for this session. I'm very excited to have Brienna Herold here. I will introduce her. She is a data engineer whose career has spanned multiple fields. She worked at the National Institute of Health, or the NIH, researching cancer drug development. She studied disaster risk reduction management in Bonn, Germany. She published her first author research analyzing autism in artificial intelligence. Most recently, Bri worked at the New York Times, managing data pipelines, and she also co-founded Signs of Good Food, a social media brand combining sign language and good food. Her talk will focus on her career pathway that was not a straight A to B. She will share research across academia and industry. Thank you for being here, Brienna.
Thank you so much for inviting me here to present today. Wonderful introduction. Thank you, Jenna. Hello, my name is Brienna Herold. You can call me Bri. I am a data engineer, and my career has been very nonlinear, which also means that I've done a lot of interesting research along the way. My goal today, whether you're a student, faculty, whether you work, whether you're at home watching this, whatever it is, I hope you learn something about data science and food and what it's like to navigate data as a deaf person. So, this diagram shows how I thought my career would go. I start with my undergraduate degree, go to grad school, get a job, teach, and then retire. That's it, A to Z. But it ended up being a little bit more complicated than that. The orange dots in this diagram are education, and the white dots are research or work experiences that I've had. And all of this can be broken down into 3 different eras. So first, I'll start with my Pipette era. That's where I ran experiments, I did data analysis, and then I'll talk about my pipeline era. That's where I transitioned from wet lab work to dry lab work, where everything was on the computer, building pipelines, analyzing data, running experiments, all on the computer. And then I'll talk about my piping bag era. And that's where I'm starting to combine both data in different areas related to food. So I'll start with a little bit about my background. I'm from rural Wisconsin. I was born deaf to hearing parents. And I also have an older sister who's hearing. But my entire family learned sign language to be able to communicate with me, and they all sign very well. I was mainstreamed for most of my life, and usually I was the only deaf student in my school district for many years, until I transferred to Metro School for the Deaf, which is in St. Paul, Minnesota, and that was when I was 15 years old. When I was 9 years old, I got a cochlear implant. I went through the surgery. I don't use the CI anymore, but I did have the surgery. And after the surgery, my aunt asked me, she said, "You just had a huge surgery. They cut open your head." And she offered to get me anything I wanted, just name it. And I think she envisioned that I would ask for maybe a book series, maybe a lava lamp, something simple, cool like that. But what I really wanted was my own computer. Now, at that time, that was in 2001, so it wasn't very common to have your own computer at home, even at 9 years old. So my mom talked with my aunt, and my mom actually thought at the same time that it would be cool for herself to have a computer too. So we ended up, my mom, my sister, and I each got our own computers at that time. And that really changed my future trajectory because I learned how to code during that time. I learned the basics, HTML and CSS, just to be able to make pretty websites for myself. Back then, there were no coding classes in school. There was nothing like that. We had keyboarding class in school, which is really just where you learned how to type, and that was it. Maybe you also learned how to use Word. So I didn't really know about the possibilities with data yet. That wasn't until I graduated high school and went to Gallaudet for my undergraduate. At that time, I didn't consider computer science as a career, so I majored in biology. So here at Gallaudet, that's where my pipette era began. This is a picture from my first research job at Gallaudet. The lab was directed by Dr. Derek Braun, who's on the left, with staff assistant Beth Kraft. This was the very first research lab that was designed with deaf space and deaf architecture in mind. So we had good sightlines throughout the lab, rather than a traditional wet lab where there's typically shelves in the middle of the bench, so you can't see what other people are doing. Here, instead of that, we were able to have clear sightlines throughout the lab so that we could easily communicate with each other. We typically had summer programs that ran, so people would come and go, but I worked for 3 full years and 2 summers in this lab. In that lab at Gallaudet, we researched this question. So mutations to the connexin 26 are relatively common, and it's a common cause of genetic deafness worldwide. It's actually why I'm personally deaf myself, is because of the connexin 26 mutation. What's interesting is that most diseases are caused by mutations, and most of those mutations are pretty rare, but the connexin 26 mutation is relatively common compared to others, and random chance doesn't explain its incidence. So what caused it to spread so widely around the world, and why is it relatively common? So we studied that by looking back in genetic history to where the mutation first emerged. So this is the type of work that we did. There are several different common mutations for the connexin 26 gene, and we studied 35 delta G. We found that this mutation, we used both biology methods and data analysis methods. And we found that this mutation first emerged about 10,000 years ago in modern-day Turkey. And from there, it spread to the European area and then to the Americas. But that still doesn't explain why it is so common. One possible explanation is this: dysentery. Dysentery is caused by a bacteria called Shigella, which is spread through contaminated drinking water. When you get dysentery, it causes bloody diarrhea, and it is very deadly. Many people died from it. Actually, more soldiers died in the Civil War from dysentery than they did from actual battles. But lab studies have shown that the Shigella bacteria that causes dysentery is actually may be affected by the connexin 26 mutation. So the connexin 26 mutation may give a person additional resistance to infection with the Shigella bacteria. But that's still an open question. It's only a hypothesis for now. Dr. Braun gives a very interesting presentation about this that's available on YouTube. So if you're curious to learn more, check it out. Now, while I was doing my research at Gallaudet, at the same time, I was also being researched on. So Dr. Braun co-wrote this paper a few years after I graduated, and I am one of the data points mentioned in the article. This was the first quantitative study that showed that deaf students, when they are mentored by culturally competent mentors who are either themselves deaf or understand deaf culture and communication, then the quality of mentoring will be better. which seems like common sense to us, but it's good to finally have data and evidence to prove that. That helps shape policy and get grants to expand more programs like this. The quality of training is very important, and it can change a student's future pathway. That's why I mentioned I don't know where I would be without the experiences I had at Gallaudet. So that lab experience, along with the deaf STEM community at that time, this was around 2010 and 2011, started having more discussions about STEM in ASL and growing the number of vocabulary items in ASL that can represent complex STEM terminology. This is one article that was published about that work during that time. And this is another quote from the article that Dr. Braun published that gives a little bit different perspective on that conversation. That conversation usually talks about what gaps there are in ASL, that ASL doesn't have enough vocabulary for STEM or science or engineering, that we need to create more signs because of this gap. But ASL is also very powerful, and we don't always talk about how rich how rich the features of ASL are, so that we can use classifiers and space and spatial relationships to express all of these different concepts. In a linear spoken language like English, for example, the word receptor, if you want to describe something that binds to a receptor, it's very clear and easy to do that in ASL in a way that makes sense. But in English, you have to understand different levels of terminology and different vocabulary. That spatial relationship isn't built into English, but it is there in ASL. And that was a huge benefit for me and other deaf students learning science. In this lab, I was also exposed to Python programming. Dr. Braun fortunately had a background in computer science already. So he would email me with small problems and small projects for me to do for fun so that I could learn how to do Python coding. I was also able to take a few programming classes, and I took discrete math. I really encourage anyone who's interested in learning programming, even if you already know programming, to take discrete math. It's a logical way that computers represent and understand materials and information. So taking discrete math will help you code better and help you understand computer science better. So I really enjoyed working on all of these problems to learn programming. After I graduated from Gallaudet, Dr. Braun also helped me find my first job at NIH. He referred me to the Cancer Research Lab, where I went to work for a few years after I graduated. This is a picture of me doing pipetting work. The research that I did at NIH was aiming to answer this question: how can we treat chronic pain directly instead of masking it with opioids? Of course, we have an opioid crisis going on right now, so this is an important question to investigate. And the answer to this question is actually tied back to hot peppers. There's a specific chemical in hot peppers called capsaicin. And when you eat a hot pepper, your body responds to it by sensing spice, and that's caused by that chemical capsaicin. Now, how does that work on a molecular level? Your nerve cells have a specific receptor called TRVP1. And capsaicin from a hot pepper, that molecule comes through your body, binds to that receptor, and that's what triggers your brain to recognize that you've eaten something spicy. The first person who proved that this receptor, TRPV1, exists is this man pictured on screen, Dr. Peter Blumberg. He was my mentor at NIH when I worked there. He's hearing, but he knew ASL, and so we could have direct conversations and communicate directly. A fun fact is that he also invented spicy birdseed. Spicy birdseed is coated with capsaicin, and it works very well because squirrels and other mammals will eat it and then taste spicy, taste like it's burning, so they won't come back and eat it again, where birds can eat it and they're not bothered by it. Their TRPV1 receptor is a slightly different shape, so the capsaicin molecule can't bind to it the same way that it does in mammals. It does bind, but it doesn't trigger the same sensation of spicy or heat. It just tastes normal to them. So in birds, the TPRV1 receptor is not worthless. It does still respond to heat, but our receptors respond to both heat and capsation. Birds only respond to heat. So the TRPV1 is kind of like your body's damage alarm. It will send a signal letting you know if you've twisted your ankle, or if you have arthritis, or if there's a cancer tumor pressing on a nerve that causes inflammation. That inflammatory area becomes warm, and that's because the TRPV1 receptors are triggering your body with that immune response. Now, unfortunately, if you have chronic pain, those signals will never stop being sent to your brain to trigger that pain sensation. Taking opioids to help does help, but unfortunately, obviously causes other problems too. This picture shows the approach that we took. If you remember, heat and capsation bind to the TRPV1 receptor, and they produce that biological response, telling your brain that you're feeling burning or heat or pain. That's what we call an agonist. Now, there's another group called antagonists, which means it binds to the same receptor, but it doesn't send a signal. Nothing happens. You can imagine a door with a keyhole. If you have the right key, you can put it in the keyhole and open the door. That's an agonist. But if you have a different key that's not right, you can put it in the hole, but then the door still won't open, and that's called an antagonist. And now the door is stuck. You can't go through it. You can't use the right key to open it. We were exploring different structures that would act as an antagonist with the TRPV1 receptor, with the goal of avoiding painkillers or other medication, but also stopping chronic pain sensations. I worked on this for several years, and we published a good amount of papers. There are several other authors on these papers. The PI, Dr. Bloomberg, frequently did hire deaf scientists, so there are other deaf co-authors here too. It was nice to be in a signing environment where we could all do research together every day. At one point, there were actually 6 of us deaf scientists working in the lab, which was more than half of the total lab members. One of the things that happened during downtime, while you're working in a wet lab, there is a lot of downtime where you're waiting for cells to grow, you're waiting for experiments to run, you're waiting for the centrifuge machine to finish working. So I again decided to learn how to code in a more formal way, and these are some of the resources that I used. And then I went to RIT for grad school.
Thus begins my pipeline era, where data flows through a pipeline, which is really just a series of tasks you perform on the data. In 2017, I began my master's degree in computer science. At the same time, I entered the Bridges to the Doctorate program. It is an NIH-funded program designed at NTID to help deaf students transition from master's programs into PhD programs in STEM and biomedical fields through viable mentoring. So it combined RIT and University of Rochester, and it was specifically designed to address deaf students' gap through the appropriate mentorship. Unfortunately, the program closed due to federal cuts. I really hope they can get something like that back. I studied flu vaccine predictions. I went to the United Nations in Bonn to do research disaster preparedness for deaf communities. I built recommendations using systems using GPT-1 for astrophysicists. Don't believe everything you see. I graduated from RIT in 2019, but I never turned in my thesis. I told them I would finish it over the summer, and I thought August, I'll hand it in, but then I went to Germany to work on disaster risk reduction at the United Nations. Then I came back to the US that fall and began my PhD in computer science at the University of Minnesota. So my MS floated away. And that's where my pathway became nonlinear. I worked in a research lab, the Knights Lab at University of Minnesota. I connected this back to my undergrad research with Connexin 26, where it is still an open reason why Connexin 26 is so prevalent. And my mentor, Dr. Knight, was interested in this and brought me into his lab where we focused on microbiome research. So it was partly a wet lab again because people would send in their feces through the mail and we'd extract bacterial DNA. It connected with my previous research. So we would centrifuge their samples, extract bacterial DNA, and try to figure out what your gut health looked like. Unfortunately, COVID hit. The wet lab closed, so I quit that PhD and went back to RIT to start my second PhD, which I could do remotely. I worked on this question. Is there autism in artificial intelligence? Or does AI view deaf people as different in any way, or is it completely equitable? I suspected that there was autism, so that was my research question. But I made a mistake. I chose a mentor who was not in my department, and I would never do that again. If I pursue my third PhD, I would make sure that we worked in the same lab together. Your PhD is specifically designed to be a mentored process. I got a mentor, but they weren't in the same lab as I was. So that made it very difficult to get guidance and project ideas when you and your mentor have different research backgrounds and different research interests. So I struggled for a year and a half, and then I decided to leave. I left academia and went into industry. I started working for a startup in San Francisco called Netlify. This is a web platform where you can build your own websites. So back to my younger days when I was learning how to code, I now worked in a company making websites. I didn't let go of my research. I still had that question about autism within AI. I wasn't in school anymore, but I'm an example to prove you can do research during your life, whether you're in college or not. So I stopped by Gallaudet University and I reached out to Raja Kushalnagar. He's a professor and a director of their HCI program. I asked if I could bring my research to campus and collaborate with him and co-author a paper. He generously agreed to help and even secured funding for me to come on as a research associate while I was finishing the research. That paper was unique because I published it while being outside of academia. The next place I was able to do research was within the New York Times. I worked there as a software engineer managing data pipelines. So if you read an article, check a recipe on the New York Times cooking app, or played one of their games, we were recording your behavior. All of that data flowed to me and my team. And our job was to process and model it so analysts could use it, because there was a lot of it. We wanted to make sure that the data is there, contained no gaps, and we built appropriate models so users could pull out the data that they needed. There were literally terabytes of data here. One thing I really appreciated about working at the New York Times was how they gave you a lot of freedom to explore their data in your own time. Every Friday, they would have hackathons, and we were allowed to experiment and build and play with new ideas. Many engineering teams are like this. You can train your app on their full archives, which is what I did. This is one of the fun projects I made. It's a time traveling game. So I helped with some other people working together to digitize the entire New York Times archive. Then I trained an AI to generate trivia questions about what happened at any day during history. And it would automatically generate questions and link readers to the original articles so they could read them. It started on July 21st, and it opened with random questions. I worked on this research project. This was my thesis at RIT, and I finally finished that thesis while I worked at the New York Times. New York Times was very generous in giving me time to work on my thesis. I ended up using the New York Times archives to analyze how people talked about deaf and deafness in journalism over time and how that has changed. This is just one small portion of my thesis and the data that I analyzed. Change in meaning over time for words is called semantic drift. And word embeddings, or a way of representing words as numbers, make it easy to analyze this because you can look at the same word's embedding and see if it has moved in this compressed 2D space. So some words are closer to other words, and they're more closely correlated. So in the 1900s, you saw the word deaf with a lot of physical disability words. It was fairly straightforward in meaning at that time. Later on in the 1900s, deafness moved in its semantic meaning, and now it was seated more closely with words like education and grammar, asylum, tutoring. By the 1990s, the word had moved into territory that involved more associations with communication and technology. That's the kind of research that uses data to analyze how language has shifted over time, which means how human beings' thinking has shifted over time. And now we reach my piping bag era. I co-founded Signs of Good Food with my partner, David Uzel. It's a personal brand that combines good food and sign language. We bring other experts to talk about topics related to food in sign language. I manage the social media, so I study the data and performance of each post to figure out what works and how to improve our processes. I found that there's nothing quite like content creation to force you to get better at communicating ideas clearly. We're still learning how to make better content that people want to watch. I also coded this map, which shows over 150 deaf-owned or deaf-staffed restaurants around the world. It's an open source map, which means anyone can come in and update it with moderation, of course. I also built a recipe database. It was surprisingly challenging to do this. It's hard to make it accurate because of all the different ingredients, so it was a very interesting data problem to tackle. We wanted the information to upload a recipe, and I would like automatic nutritional information to appear with it. With my data work, the goal is always the same. Use software engineering and data engineering to save time, surface new information, make our lives easier, healthier, and more interesting. So this is a visual of the technology I have worked with. I call it the modern data stack in the AI era. It's something that all of these projects have in common. It's a scientific approach. It involves statistics, knowing how to get up to speed in a specific domain knowledge, from biology to food to medicine, and it's different than AI autism research, but in the same way, you're working with data and data sets. So if you're just starting out, I would recommend taking discrete mathematics, learning SQL, and then Python. Right now, I'm freelancing as a software engineer, and I'm open to work, but who knows what's next? Maybe I'll go for my third PhD. Maybe I'll open a restaurant with signs of good food. I don't know yet, but that's the nice thing about data. The skills you develop working with it are some of the most transferable ones out there.